Hello everyone! Today in this video we are going to talk about the steroid-resistant nephrotic syndrome according to where their proteins function in the podocyte. So instead of memorizing one long and confusing list of genes responsible for this kind of steroid-resistant nephrotic syndrome, we will divide the genes into cellular pathways. And throughout this presentation, you would see that the red color would represent recessive inheritance, while the blue one would represent the dominant inheritance. So the goal is to create a simple mental map that is easier to review and to remember for you guys. So if you are ready, let's kick off. First of all, I would like to begin at the glomerular filtration barrier, especially the slit membrane and the area where the podocyte attaches to the glomerular basement membrane. So let's begin with the slit membrane. The recessive genes are CRB2, CD2AP, MPHS1, MPHS2, and FAT1, and DGKE. These are colored in red, which means they are recessive, and the only dominant gene here is in this group is the TRPC6. And next, we have at the podocyte cell surface, we have the WDR73, PTPRO, and EMP2, Cubbon, and CFH. These are all recessive in this figure because they are all colored in red. And then just beside it, we have the integrin-laminin interface. We have the genes ITGA3, ITGB4, and LAMP2, and these are also recessive genes. So the memory rule for this slide is that the slit membrane has 6 red genes and 1 blue exception, which is TRPC6. So 6 red, TRPC6. I think using 6 to memorize these would be easier for you. And then we have the surface and the attachment genes, which are all colored in red and are recessive genes to remember. And then we would move to the podocyte's actin cytoskeleton, which forms the structural framework of the food processes. First, we will begin at the actin-binding genes. The recessive genes are MYO1E, DAM2, IVIL, and PLCE1. The dominant genes are ACTN4, MYH9, INF2, and NLN. If we look closely here, this gives us a balanced pattern. For example, we have 4 recessive and 4 dominant actin-binding genes. So 4 red and 4 blue. And always with the actin cytoskeleton, we have the actin regulation genes, which might give nephrotic syndrome by several genes like ARGDIA and KANK1, KANK2, and KANK4, ITSN1 and ITSN2, MAGI2, and TNS2, and DLC1, CDK20, and NOS1AP. The single dominant exception in this pathway is the RGAP24, colored in blue, which is dominant gene. So if we want to remember these genes, we would say that we have actin-binding proteins and actin regulation proteins. In the first one, in the actin-binding proteins, we have 4 recessive genes and 4 dominant genes. On the other hand, the actin regulation proteins, we have 1 dominant gene, which is RGAP24, and for the rest of the genes, we have recessive genes. I would like to notice that these genes of the regulation They are known for their non-resistance to steroids. I mean that if we have a nephrotic syndrome and we might diagnose a mutation in the actin regulation gene, your nephrotic syndrome is highly likely to respond to steroids. So give it a try. And then we're going to move inside the podocyte and follow a simple sequence. We have the nucleus, we have the nuclear pores, and we have the endosomal trafficking. For the first one, the nuclear control, we have WT gene, LMXB1 and TRIM8 and SMARCL1, which is the only recessive gene in the nuclear control. And then just beside it, we have the nuclear pores, which contain recessive genes like NUP107, NUP85, NUP133, NUP93, NUP205, and XPO5. And finally, we have the endosomal trafficking genes, which are ANKPhi1, GAPVD1, and TBC and D8B. These also are recessive genes. So for memory, we have the nuclear control, which is mainly blue, with one recessive gene. We have the nuclear pores and the endosomal trafficking. They are both all red. And then we would move to the last slide, and we would talk about the other genes. We would talk about the CoQ10 biosynthesis, which is found in the mitochondria. And we would talk about the tRNA modification COPS complex, and we are talking about the metabolism of S1P, and we would talk about the genes inside the lysosome. First of all, for coenzyme Q10 biosynthesis and the mitochondrial function, the genes are ADCK4 and CoQ6, CoQ2, and PDSS2 and MTTL1. For the tRNA modification through the COPS complex, the genes are OSCAP, TP53RK, TPRKB, and LAG3. The only gene involved in the S1P metabolisms of lipid is SGPL1, and the lysosome gene is SCARB2. All these genes, as you can see, are colored in red, so they are recessive genes, which makes it easier for you to remember. So they're all colored in green, they are recessive. And now, guys, we can now simplify the entire figure by memorizing the only blue-colored gene. So we have only 9 dominant blue-colored genes, and they are the exceptions because all other genes are colored in red. For example, we have in the slip membrane, we have the TRPC6, and the actin binding, we have ACTN4, INF2, MYH8NL here, and we have in the nuclear podocyte, we have the WT1, LMX1B, and TRIM8. In the actin regulation, we have the RGAP24. So there is a way, a mnemonic code that you can use in order to remember these exception genes which are dominant. It is T-AMIA LWT. T stands for TRPC6. AMIA stands for ACTN4 MYH8 A, INF2, NL, and A, RGAP24, and LWT, LMX1B, WT1, TRIM 8. So memorize these 9 blue genes, and every other gene in the figure is recessive. I think this is a very good way to remember, and it would make it easy for you to memorize. And like this, you can remember the remaining genes in this particular figure as red and recessive, as it is mentioned here. I think we came to the end of this video. I hope it was a beneficial one. And see you next time, guys.